Hi everyone, this is Dr.T.SUMATHI, this is DG and Research Department of Zoology, A.D.M. College for Women, Nagapattinam. In this PPT, we have to know about the subject, very interesting subject, cell biology. First, we know about what is meant by cell, why we have to study about cell, what is the role of cell in our body. Normally, cell biology is the study of cell structure and function, and it revolves around the concept of the cell. Cell is a fundamental unit of life. It also focusing on the cell. It permits and detects to understanding of the tissues, organism, and everything on the cell component. This is a basic structure of human cell. These human cells, body cells also have the, very well about the outer membrane, that is called cell membrane, and also it content of the material, protein material, that is called cytoplasm. The plasma membrane filled with cytoplasm, and also it have the center part, that is called nucleus. Nucleus also have the nucleolus. That nucleus function is, main function is it carrying the genetic material. You know the genetic material, that is a thread-like structure, that is called chromosome. The chromosomes also have the gene. The gene also transferring the genetic material from one generation to next generation. So the genetic material in the nucleus also very, very important. Another cell organelles also ribosomes, lysosome, Golgi apparatus, Golgi vesicles, peroxisome, smooth endoplasmic reticulum, and rough endoplasmic reticulum, and it has some chromatin substance, that is a chromatin also plural of chromosome, centrosomes, everything also. What is the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum? The rough endoplasmic reticulums and the ribosome is present in the endoplasm, that is called rough endoplasmic reticulum. Without ribosome, that is called smooth endoplasmic reticulum. And it has so many vacuoles. That vacuoles also one of the cell organelles. The nucleus also, second part, it has the nucleolus. That is very important. The nucleolus function in the all cell functionings. The cells are the structural and functional, and also it is a biological unit of all living beings. Biological unit means that is a living things only we can see the cells, and also cell functions. Normal, all the cells can replicate itself independently. Replicate means they have to self-reproduce. That is a 2 cells is formation of 4 cells, after 8 cells, next 16 cells and 32 cells, and also formation of blastula, gastrula, morula stages. That is, we know about the developmental biology also. That is a replicate itself independently. Hence, they are known as the building block of life. Each cell contains a fluid that is called cytoplasm, which enclosed by the membrane. That membrane is called plasma membrane. And also it presents the cytoplasm or several biomolecules, that is like protein, nucleic acid, and lipids. Everything also present in the cytoplasm. Normally, protein also very, very important in our body, because the body builders also protein. All our body also made up of protein. Protein gives the energy, everything. The nucleic acid is nothing but a DNA that is present in the nucleus, and a lipid particle all over body. Our body also have the lipid things. Moreover, the cellular structures is called the cell organelles. It is suspended in the cytoplasm. After that, we have to know about who discovered the cell. The scientist is discovering the cell. The scientist is called Robert Hooke. He discovered the cell in 1665. Normally, first the Robert Hooke observed the piece of bottle cork under the compound microscope. That time also noticed one of the minute structure that reminded of the small rooms. Already I told you, we can see the biological things. After that, they have to identify that is a living being. First, they have to focus on the non-living. Consequently, he named this one of the rooms. He don't know the cell, the word rooms. However, his compound microscope had the limited magnification, and hence he could not see any details in the structures also. Owing to this limitation, the Hooke concluded that these were non-living entities. After that identification of Robert Hooke, later one of the scientists identifying this one observed the cell, Anton von Leeuwenhoek, observed under the compound microscope with high magnification. Why the magnification is high resolution? That time only we can see the internal structure of the cells. This time he had noticed all the cells exhibited some form of the movement. That movement is always called motility. As a result, the Leeuwenhoek concluded something they have to alive in the microscope, that is entitled in the alive in condition, living in condition, evenly after post of other observation. It is he named as animalcules. That time also Anton von Leeuwenhoek don't know the name cells. That time they have to identifying animalcule. In 1883, one of the scientists, Robert Brown, the Scotch botanist, provided the very first insight into the cell structure. He was able to describe the nucleus is present in the cell in the orchid flower. After that, the following the various essential characters, and also it provides the supporting the body, and they have to identifying all the cell interior, everything of the some of the nucleus major role. On the side by side they have to identify all the scientists. Normally the nucleus contains the major cell organelles, there is a genetic information that is necessary for the reproduction and the cell growth also. Why these reproductive cells? Already you know the chromosomes. XX chromosome is a male, XY chromosomes is a female. That is a reproductive cells, also very important. Each and every cell have the one nucleus and the membrane bound the organelles in the cytoplasm. Normally we have to know about some cell organelles. Already I have explained so many cell organelles is present in the cell. That is called, this one is a mitochondria. The mitochondria is very important in the cell. Why? Because that is all the energy, electron transport system, everything also formed in the mitochondria. This mitochondria is a double membrane. It bounds mainly in the response of the energy transaction within the cell, and it is a vital role and survival of the cells. After that, the cell formation of electron transport system, it gives the energy. Then normally ATP molecules formations in the within the mitochondria. After this mitochondria also transfer from mother to offspring also. That's one of the very important. And lysosome. Lysosome, one of the cell organelle. It digested, the role is a digestion. That is unwanted material in the cells. It is a digester, and it have the two role. Lytic function, that is a lysis function. One is a phagocytosis, another one is a pinocytosis. Phagocytosis means cell eating. Pinocytosis means cell drinking. They do the function. After that, in the unwanted things, they have to removing from the cell. After, one of the important one is the endoplasmic reticulum. It plays a significant role in the internal organization. What is the role of endoplasmic reticulum? Already I told you, rough endoplasmic reticulum. Another one is a smooth endoplasmic reticulum. The presence of ribosome in the cells, we have to differentiate these synthesizing, and also it used to directing and the sorting of the appropriate location in the cells in the body. We have to know about the substance, one of the substance, colloidal substance, that is called the cytoplasm. The cytoplasm is present both plant and animal cells also. There are jelly-like substances. It is a colloidal substances found between the cell membrane and the nucleus. Normally, they are mainly composed of water and organic and inorganic component also. The cytoplasm is one of the essential component of the cell and all the cell organelles. All the cell organelles also embedded in the cytoplasm. These cell organelles contain the enzymes mainly responsible for controlling all the metabolic activity and also taking place within the cell and the site of most chemical reaction within the cells also. And the plasma membrane. The plasma membrane is also termed as cell membrane or cytoplasmic membrane. It is a selectively permeable membrane of the cell which is composed of lipid bilayer and also protein. These plasma membranes also it present both function on the animals and the plant cells as such a cytoplasm. And the plasma membrane function is a selectively permeable membrane. It also permitting the entry of the selective materials according to the requirement. What are the selective materials means? In case any one of the disease affecting the body, that time also barrier to occurs in the cell. It is an animal cell. The cell membrane functions by the providing shape and protect the inner content of the cell. It is very important to the plasma membrane. And also one of the center part that is called a nucleus. The nucleus is a double membrane. The organelles found in the all eukaryotic cells. It is the largest organelle. It also controlling the center of all cellular activities like DNA, RNA, everything also. It also occurs in a DNA in a double helix structure. The DNA also contains a gene. So it acts as a genetic material. By this structure, the nucleus is a dark round surrounded by the nuclear membrane. Nucleus also covered by the nuclear membrane. Normal pores of the membrane like cell membrane form between the cytoplasm and the nucleus. Within the nucleus, these have the tiny spherical bodies. It is called nucleolus. These nucleolus it also carries an essential structure that is called chromosomes. That chromosome also carrying the genes, everything also. Normally chromosome also important in the genetic material. After that, that is called endoplasmic reticulum. That endoplasmic reticulum is a network of membrane that is filled with the fluid. They are transport the system of cell. It involved in the transporting material throughout the cell. There are two different type of endoplasmic reticulum. One is a rough, another one is a smooth endoplasmic reticulum. Normally, rough endoplasmic reticulum composed of cisternae, tubules and vesicles, which are found throughout the cell and involved in the protein manufacture. The smooth endoplasmic reticulum, they are the storage of organelle. It is associated with the production of lipid, steroid and also responsible for the detoxifying of the cell. After that, already I told you the mitochondria, that is a vital role in the mitochondria. It is otherwise called powerhouse of the cell. They produce the energy-rich molecules in the cell and also it has some function inherited maternally in the several organisms. That's very important. It is a double membrane, already I told you, it is the shape of the organelle. It found in the all eukaryotic cells, not prokaryotic cells. You know the eukaryotic means true nucleus. Another one is the important one is a plastid. Normal the plastid is a large membrane. It found in the organelle which contain the pigments. The pigments is based on the three types, normal red pigment, blue pigments, everything also. Chlorophyll is one of the pigment, that is a green pigment. Another one is a ribosomes. The ribosome is a non-membrane bound in the important of the cytoplasmic organelles. And it association with the endoplasmic reticulum. The ribosome is very important for the protein synthesis. The chain of the amino acid to formation of protein synthesis. Ribosome is one of the part to formation of protein. It is mainly composed of RNA and 1/3 of the protein and 2/3 of the RNA. The ribosome has so many types, 70S, 25S, 22S. That is a sedimentation coefficient. It has a different type of ribosome is present. After that, one of the cell organelle is a Golgi apparatus. These Golgi apparatus are also termed as Golgi complex. It is bound in the organelle. It is found at the flathead and pouches. That pouches is called cisternae. These cell organelles is primarily responsible for transporting and modifying of the cell structure and packing the protein and lipid in the targeted position. After that, the Golgi apparatus found in the cytoplasm. Normally, it represents both animals and plant cells also. Above slide also we have to know about what are the cell organelles is present in the normal cell. That is the usage of the cell organelle. That is a basic we have to know all the cell organelle. After that, in this unit we have to know about the cell fragmentations. Why we have to study the cell fragmentation? After fragmentations of the cell, we have to identify what are the cell organelles present in the tissues. So the we have to know about the cell fragmentation in your syllabus. In this method, we have to separate the subcellular component and the organelles from the tissues, and at the same time we have to know about the structure, function, molecular composition and the isolated component may be studied in this cell fragmentation. Normally, this process cell fragmentation is started homogenizing the tissues in the buffered isotonic solution. What are the material we have to use of homogenization? Morton pistil. After that, we have to identifying cell fragmentations. This is a diagram. This diagram also tell how we fractionate the cell from the tissues. We have to use of some procedure. Another one method is called centrifugation. This centrifugation is one of the method. We have to identify the concentration of the content of the molecule in the cell or any one of the mixture. Centrifugation is the process. It uses the centrifugal force. After that, we have to separate the two liquid mixture. The process, they have to migrating the larger molecule from one place to another place, and lighter molecule component is occurs migrated to the base. Normally, centrifugation the process separating the material in the suspended liquid medium. This is mainly based upon the gravity of the particle suspension. Normally, there are two type of particles occurs. That is one of the particle is a supernatant, that is a liquid part. Another one is a content is called pellet region. The centrifugal force is a proportional rotational to rate of rotator. The centrifuge consists of rotor. It is close to the refrigerator and also by the electrical motor. The centrifuge unit operation is working for separation of the some centrifugal force evolved in the any one of the techniques we have to following to separating the solution, and maybe the separating of the solution mainly depends upon the size, shape, density and the viscosity of the medium and the rotor speed. Rotor speed means one of the force in the center, after that they have to rotating. The centrifuge consists of container in which a mixture of solid, liquid and also it contains a very high speed. When the container rotator, the high speed mixture and separated constituent they have to use it, and higher specific gravity also we have to use it. And we have to know about the solid and the liquid medium of the specific gravity and thrown out the word and the greater of the force. This is the diagram, that is a laboratory centrifuge. After we have to homogenize the cell, we have to incorporate in the centrifuge tube. Then we have to close and switch on the centrifuge. They have to rotating that once. After we can see the supernatant and the pellet. This is a machines used to centrifuge. What are the principles we have to following in the centrifugation? It involves some of the sedimentation process. The principle of centrifugation technique, it is separating the particle in the liquid media, and also it is sedimentation phenomenon is suspended the material and settle out the fluid by the gravity. The particles having more than 5 micrometer and separated by the simple filtration process. The 5 micrometer or less do not sediment under the gravity. Normally the force is used to separate those particles in the gravity of the medium. What are the objectives of the centrifuge? What are the objectives we have to following the centrifuge? To separate the immiscible of the liquid and to purify the component by removing the impurities in the supernatant liquid. This centrifuge used to separate the crystalline drugs from the mother liquor. The test also emulsion and suspension of the any one of the creaming and the sedimentation and accelerated speed. We can use the objectives of the centrifuge. Then we have to know about the what is the centrifuge, what is the role of centrifuge, and also what is the force uses of the centrifuge. After centrifugation, what are the matter we have to getting, that is one is a supernatant, another one is a pellet. After we have known about the application of centrifugation. First one, production of bulk of drugs. Another one is a production of biological product. Another evaluation of suspensions and emulsions. Another one is a determination of molecular weight of the colloidal particle. Another separating of the chalk powder from the water. Another one is a removing fat from the milk to produce a skimmed milk. In market also available without fat condition, that milk also they have to use of the centrifugal force. The clarification and the stabilization of the wine particles. Another main important biopharmaceutical analysis, we have to use the drugs. After uses in the water treatment, we have to use of the centrifugal force. During water treatment, removing the water from the lettuce, after washing the salad spinner, separating all the particles from the airflow method, and then we have to using the cyclonic separation, that is the rotation of the separation. Another one of the important topic is here, is staining. Why we have to know about the staining? Any one of the tissues we have to identify, we have to use of the different type of staining. Then only we have to identify internal part of the cell structure. What are the type of staining technique? There are two type of stain method. One is a simple staining, another one is a differential staining. Simple staining we have to use of the single, that is a direct or positive staining. Another one is a indirect or negative staining. Another one is a separation into group. One is a gram staining, another one is a acid or fast staining. Another visualization of the structure, flagella stain, capsule stain, another one is a spore stain. What are the role of gram stain? The difference in the dye, retention in the depend on the such physical properties as the thickness, the density, porosity and the integrity, everything also occurs in the bacterial cell wall as it is a chemical composition. What is the gram-positive bacteria? Why we have to know about these bacteria? The gram-positive bacteria have the thick, dense, relatively non-porous walls, while the gram-negative bacteria have the thin walls surrounded by the lipid or a rich membrane. Some non-bacterial organism with thick cell wall, it have the some of the stain that is called gram-positive bacteria. These gram-positive bacteria which have the lost wall of integrity through the aging and the physical or chemical damage by the gram-negative bacteria. We have to use of the gram staining, that is staining for part procedure. Normally the specimen is mounted or heat fixed in the slide and we have to identify using some of the reagent. That reagent is called crystal violet and iodine solution and decolorizer and saffronin and water. Preferably we have to use of this staining method. This is a basic steps for gram staining method. First, microscopically we can see this gram staining. We have to apply the crystal violet on the slide and keep it on the 60 seconds. Wash the slide with the water. After we have to apply the slide, keeping the water 95% of the ethanol, and we have to keep on slide 5 to 10 seconds only. And we wash the slide, we focus the slide through microscope. First one is a crystal violet, that is a purple color, and that is developing the 60 seconds they have to develop in purple color, that is a, we have to identify gram-positive bacteria. Then we have to use of the iodine, that is a orange color, we have to use 60 seconds. We have to identify the gram-positive bacteria also. Next, we have to use as 95% of the ethanol. 5 to 10 seconds, we kept it in the 5 to 10 seconds. We have to identifying both gram-positive and gram-negative bacteria. After that, saffronin. The saffron also identifying the pink color. We have to identify both gram-positive and gram-negative bacteria. Depends upon the color, the coloring of the agent, we have to identify these particular bacteria is present in this particular one. Thank you.